Myogenic tone is a characteristic feature of small arteries and arterioles, although the mechanisms are not fully understood. I aim to explain what myogenic tone is, why it is important, and the underlying mechanisms as they are currently understood. Myo means muscle, and genic means originating from. So, myogenic tone is a tone that originates from muscle. When applied to arteries, it is a continuous baseline level of tension in the walls of the arteries that is generated from within the smooth muscle. This cross-section through an artery has been stained with hematoxylin and eosin, or H&E, to highlight cell nuclei and smooth muscle. Arteries are covered with a layer of connective tissue known as the adventitia. The innermost surface has a continuous layer of endothelial cells, although only their nuclei are seen here. In between the adventitial and endothelial layers is the medial layer made up of concentric rings of vascular smooth muscle cells. When these cells contract, the artery constricts. When the smooth muscle cells relax, the artery dilates. You can see myogenic tone develop when you change the pressure at which blood flows through an artery. As blood flows, it exerts pressure on the inner walls. When the pressure increases, the walls are pushed further out, the artery becomes wider, and there is an increase in the flow of blood through it. In small arteries, that effect is short-lived. The increased force on the artery wall stretches the smooth muscle cells, and they respond by contracting. So the artery constricts. Its purpose is to maintain blood flow in tissues at a relatively constant level, regardless of fluctuations in systemic blood pressure. Most sensors of pressure in vascular smooth muscle respond to mechanical stretch of the cell membrane. Multiple smooth muscle proteins can respond to stretch and have been proposed as sensors that initiate the pathways leading to myogenic tone. It is widely accepted that stretch-sensitive ion channels are involved. Several members of the transient receptor potential family of cation channels can sense stretch. There is direct evidence that TRPM4 and TRPC6 are involved in the myogenic response, but while they were once thought to sense stretch directly, it now looks like they are activated indirectly by signals downstream of G-protein coupled receptors. Other types of cation channels have also been proposed as stretch sensors, but their involvement in myogenic tone is less clear. Some G-protein coupled receptors have been found to activate when the membrane is stretched, even in the absence of an agonist. There is strong evidence for angiotensin I receptors in the initiation of myogenic tone. For example, myogenic tone is suppressed by angiotensin receptor antagonists like losartan in the absence of angiotensin. Integrins are transmembrane proteins that link the contractile machinery of the smooth muscle cell to the extracellular matrix. They can sense stretch in the artery wall and stiffness in the extracellular matrix. Without integrin function, myogenic tone is impaired. Matrix metalloproteinases, membrane-bound tumor necrosis factor alpha, and the tumor necrosis factor alpha receptor have all been implicated in myogenic tone. Their roles are unclear. They may have a modulatory role rather than participating in the main pathways leading to myogenic tone. This all gives a picture of multiple contributors and complex processes leading to myogenic tone. But we don't yet understand the significance of all these sensors. For example, do they operate independently, or do they link together in unknown ways? Or is there differential expression of sensors in different vascular beds, mediating different mechanisms? Regardless of what senses stretch, we do know that pressure induces myogenic tone by depolarising the vascular smooth muscle cell. That is likely achieved by non-selective cation channels like the TRPM4 and TRPC6 channels. Whatever signal causes these channels to open, the main ion that permeates them is sodium. Some cation channels, like the TRPC6, also allow calcium ions to flow through, but the main role of these channels is to mediate sodium influx and depolarize the cell membrane. This affects the activity of voltage-dependent ion channels in the membrane, especially the voltage-gated calcium channel. As depolarization spreads along the cell membrane, it opens the calcium channel, allowing calcium ions to enter the cell. Calcium entering the cell in this way triggers contraction. There is evidence that the process might be amplified by calcium-activated chloride channels. As the cytoplasmic calcium ion concentration rises, calcium can bind to and activate the chloride channel. As the channel opens, chloride ions flow out of the cell down their electrochemical gradient, and that depolarizes the membrane further. Membrane depolarization also activates voltage-gated and calcium-activated potassium channels. When these channels open, potassium ions flow out of the cell down their electrochemical gradient. That promotes membrane hyperpolarization and opposes the depolarization, thus promoting closure of voltage-gated calcium channels. This limits the amount of tone that can develop. The calcium-activated potassium channels, known as BK channels, are also activated by calcium ions, but they need a higher concentration of calcium than is usually reached in the cytoplasm. The sarcoplasmic reticulum acts as a store of calcium and releases it through ryanodine receptors, which are located next to BK channels. Calcium release occurs in bursts, known as calcium sparks, which create a microdomain with a high calcium ion concentration around the channels. Each spark activates BK channels to induce a transient flow of potassium ions out of the cell and membrane hyperpolarization to oppose the stretch-induced depolarization. This mechanism is enhanced by pressure-induced stretch of an artery. Stretch causes the production of reactive oxygen species in the artery wall, which stimulate the activity of the enzyme protein kinase G. Protein kinase G, in turn, increases the frequency of calcium sparks, BK channel activation, and membrane hyperpolarization. Loss of this mechanism has been shown to disable the negative feedback regulation of myogenic tone.